At the Leibniz-Institut for Molecular Pharmakologie, Dr. Jens-Peter von Kries and his team have established an exceptional laboratory for large-scale automatic experiments. The so-called screening unit is the main technology platform of the German Initiative for Chemical Biology, ChemBioNet. By linking biological and chemical methods, Dr. Kries and his team help to find out the function of proteins. They support academic research projects looking for new active ingredients.
Biologically
active substances have unique characteristics that make them a valuable tool for fundamental research. Since the projects we are supporting do not depend on a connection to the disease, in comparison to projects in the pharmaceutical industry, we can collect data on a much wider basis and therefore possibly gain surprising insights. The screening unit has a library of more than 50,000 substances. They are tested for active ingredients with the most modern technologies. For example, the screening laboratory is the only academic institution in Germany to work with a lab-on-a-chip device. Among others, it's used to look for active ingredients for treating tuberculosis and other infectious diseases. This miniature laboratory on a quartz glass chip is similar to an electronic chip, the difference being that, instead of etched electronic lines, it includes a microanalysis system. Samples are pumped over it with an electrical field being fed at the same time, so that these samples can be analyzed on a very large scale. This technology allows us to screen our entire substance collection in only 2 days. Pipette robots manage the same. They can do more than 40,000 test reactions in less than 3 days. Almost 400 pipette tips transfer the various substances to the test plates. The reactions then show the function of genes or the effect of the substances. When the tests are completed, Dr. Ronald Kuhner from the research group Drug Design analyzes the data using special programs that are also used for developing medication. Kuhner and his team support the screening unit with the automated data collection and computer-aided chemistry.
Im Mittelpunkt unserer Forschung steht die Aufklärung der Beziehung.
Finding out the relationship between chemical structures, the characteristics of the chemical connection, and the biological activity of the connection is the focus of our research. We try to develop models on the computer which better describe such structure-activity relations, identify them, and use these models to predict new, better active connections.
Neue, besser aktive Verbindungen vorherzusagen.
On the one hand, the function of the proteins that are examined in the screening department serves the search for active ingredients. On the other hand, the scientists also want to find out the cellular target of the active ingredients. Using automated microscopes, they therefore examine the communication paths of the cells. These paths are especially significant for cancer. With the help of a test in cell cultures, migration of tumor cells from the cell colonies can be observed, for example. This process corresponds to the formation of metastases.
We want
to enable academic projects to access the large-scale search for active ingredients and support them in the best way possible through know-how that is collected here. And since we also receive substance donations from chemists that are impossible to purchase, we have unique resources that are only waiting to be used here.